this is probably the practice that has fed me the most. All right. Meaning I've done a lot of work in bioretention, and I probably have my most journal articles associated with bioretention. And it was my PhD project. So bioretention is a very important, near and dear to my heart project practice. Does anyone want to guess on my second favorite? And maybe this might be my second favorite practice. My favorite practice is a constructed wetland. You know why? Because they're resilient. They're resilient. Wetlands are the most resilient practice we got. But bioretention, because it employs vegetation, is also pretty resilient. And bioretention does a lot of good when it's working. Okay, here's the first known practice of its kind. I'm going to blow through this because we will be done before 1 o'clock. Is it? Thank you. Does that look good? Okay. I'm going to go through a number of the can of bioretention. Okay, come on. Uh-oh, okay, there you are. This is the first one that I ever designed. You can see 1998. There's the building. There's my tree. It was a very nice practice. And then it flooded, and that's my tree right over there. Underwater, the thing was dead. We built it and made it back. So you can see it's a pretty attractive practice, right? We started doing a bunch of research on them. This is my PhD. In fact, these are my three projects for my PhD, and there I am as a PhD candidate trying to figure out what to do. A little bit of dig in action. All right. So this is where I did my research. And what I'm going to do now is I'm going to show you all a series of bioretention. Here's one where it's been used to treat residential runoff. This is some very rich person's house. That is serious richness right there. And they have a big rain garden in front. This is not a rich person's house. This was my house right there. That's my house. There I am. We love American college football in the South, in the southern part of the United States, as well as the Midwest. College football, meaning American football, is super popular. We'll get 100,000, 110,000 people show up and watch every Saturday. It's a bizarre phenomenon for most people from overseas. Like, what's going on here? All right. Well, we were watching a football game. My university's colors were red, so you can see where we're. And built this rain garden. All right. And then the plants bloomed and I had honeybees. I felt really good that day. I had honeybees. In fact, you can design rain gardens or bioretention cells to specifically attract fauna that you like, especially butterflies, because who here doesn't like a butterfly? If you see someone who doesn't like a butterfly, that person's probably has a very hard heart, okay? If you can't like a butterfly, that person probably needs extra love and attention. All right. Just FYI. You'll see rain gardens be integrated into neighborhoods, into streetscapes. Here's one, a very pretty example of a streetscape in Seattle. Here's another one. This is actually in Copenhagen. I talked about this actually on Monday. Here's one in Topeka, Kansas, in the heart of the United States. They took that street and they turned it into something much more attractive. You can see all the rain gardens there. Looks really good. Too bad. The Wizard of Oz. This is Kansas, where the Wizard of Oz took place. All right. This is in New Zealand. That's a very excited rain garden. Okay. Here's one in Asheville. This is like European style football. Here's the football grounds. And there's a bioretention area that's shaped like a football that people drive around as a roundabout. That's kind of cool, right? Here's one that we designed. Here's your parking lot, a big median. Inside that parking lot median is a stormwater treatment device. I think it looks pretty good. Again, that's one that I designed with my team. These boxes you see here, that means we're monitoring it. All right. Here's one where the rain garden is built on the side of a building. So the rooftop leaders come down and it discharges some of the rooftop water into this garden area on the side of a building. All right. Here's one at NC State campus. Again, side of a building, next to the building. It's a pretty attractive design. So you get an idea that these things can be good looking and they are garden-like areas that accept water. This is our big library. Y'all have the Mahatma Gandhi Library. If you know anything about that, that's my last name. You might be thinking, so Dr. Hunt, did you give money to this library? The answer is no. But a former governor of our state did. And that is the name of the library for him. So this is the reading room in the library, right? And next to this big glass window, it's nice. And outside of the reading room, what do you see here? Those are all rain gardens, right? All bioretention cells. So they literally integrated the inside space. This is where architects can do some pretty cool things, all right? Engineers usually have to make it work. Well, the architects, basically the interior architect, center architect, worked with the landscape architect to create this really neat green space for the students and the faculty, and from my perspective, does stormwater management. All right, here's one in the heart of North Carolina's largest city. You would not confuse the largest city in North Carolina with the largest city in India. Trust me, there's a big difference. Y'all cities are a lot bigger, all right? But here's one that's been installed in Charlotte, and it looks pretty nice. You know, ultra urban area looks pretty nice. Nothing to remember, these are living systems. Here is the rain garden that I studied actually after it was first built in 2001. Here it is 10 years later. I'd love to show you the picture of it now, but guess what? In 25 years, it's been taken over and something else has been put in its place. But it actually well 15 years in. I monitored it. Here's one where the Walmart, which is a big shopping store we have at home, here it is early, and then here it is later. What do you observe? That these systems evolve. Why? Because they are vegetated, they are biological systems, and if you give something 10 years, 15 years, the plants are going to grow, and honestly, some of those plants are going to die, all right? And so making sure that the property owner understands that you're looking at an evolving system is super important, all right? So the take home from all of that is there's lots of different places that you can use bioretention. You can use them in ultra urban areas. Yeah, you can use them in ultra urban areas. It is very important that you're draining either very stable areas or impermeable surfaces. So rooftops, car parks, roadways. That's where retention works. All right. All right. Now, what I want to do is talk a little bit about how they're designed. That's my youngest daughter. She's much older now, very funny now. She's a funny girl. So basically, when it rains, the water comes in, flows in through the media, goes out an under drain, soaks into the media, as well as exfiltrates, as well as evapotranspires. You guys ready? Everybody up. Everybody up. You're going to remember this one thing. You've done it once, and you're going to do it again. All right? Instead of coming up like this, the water is actually coming in. So you got to do the water's coming in. It's into your bioretention cell. It can go down and out all the way. Okay? It can go down and then out of pipe. This is almost like a yoga move, right? Yep. It can evapotranspire. And if it rains a whole lot, it comes in and then just keeps going. There you go. There isn't another practice that I have shared with you that does this and this as well as bioretention. So one of you asked about like urban areas and trying to improve the hydrology of ultra urban areas. You're looking at the best practice we got. What are green roofs good at? If a green roof does this, there's a problem, right? Green roofs do that. But bioretention does this and that, all right? Wetlands, ponds do a little bit of that. Permeable pavement doesn't really do any of this, but it does that. You see what I'm saying? So bioretention is the one practice that you get evapotranspiration and potentially good infiltration. All right, good job standing up, everybody. You'll remember this workshop, this week-long workshop, for the times I had you do your stretches, all right? Now, one of the tricks behind bioretention is that if you're going to get good infiltration, you've got to have some separation from the bottom of your bioretention area to the top of your seasonally high water table. I'm going to repeat that again, because this matters here. If you're going to get good infiltration, you got to make sure the seasonally high water table isn't up near the surface. I'm not familiar with Mumbai. We were talking about Mumbai earlier. I have a desire to go to Mumbai. And great part, you have great architecture there. I happen to like architecture. If any of you happen to come to America, first of all of you are invited by me. Okay, I mean, other people have to say yes as well, but when you come and you like architecture, go to Chicago or go to New York City, the two best places for architecture in the United States. Chicago is, both of them are fabulous. Chicago is more manageable, but if you're from India and you can handle Mumbai or Delhi, then New York City won't be a problem for you either, all right? Now, but if your water tables aren't not up near the surface, this might be a good practice for you, okay? I just know that Roorkee is, the water is kind of near the surface in some places. Could I use a bioretention near the Thomason building? I think I could. Because it's up on a hill, all right? But you've got to make sure you've got some separation from the bottom of your cut to the top of your seasonal high water table. I've got an example that I would normally show you to illustrate that, but I'm just going to blow through that right now, okay? Just for time's sake. All right? I will say this, that in some cases, if you're not really worried about improving or infiltrating water, you're not worried about having a significant change to the hydrology, then you can cheat a little bit and have your water table get close to the bottom of your bioretention cell. You won't have water soaking out the bottom. You won't be exfiltrating. All the water will actually soak in and drain out the underdrain. But if that's what the system you have, it can be very good at removing certain pollutants. Here's E. coli concentrations. Those are some pretty good numbers, y'all, right? We've gone from ten. So, and your body, well, doing right. Tell me it's not. Please don't be gone. When the pathogens are hitchhiking with sediment, the sediment's trapped by filtration. Where's the pathogens trapped at? At the surface, right? Then what's that pathogen exposed to? Sunlight, evapotranspiration, desiccation. All these things kill pathogens, right? And so if the pollutant removal that you're targeting happens to be confined to the top surface, then it probably doesn't matter what's happening, a meter beneath your foot. Okay? Just be mindful of that. So often people will get all hung up on design standards, which I understand, okay? But then you get lost in the fact that what you're trying to do, well, you might be able to bend some of those design standards and still get good performance. So how deep should you allow water to pond above the bioretention surface? And the answer is your water quality volume would ideally pond about 30 centimeters above your surface, your water quality volume. Now that which associated with 35 millimeters, okay? However, you could have perhaps a half a meter, all right, or even more, 0.6, maybe even 0.7 meters, that all the water can fill up and drain out if you're trying to mitigate peak flows, okay? So this practice does allow you to mitigate peak flows. And when I remember from the first presentation, the targets they call it a class 1 city was the 5-year storm. My hunch is that bioretention cells can be good at 5 years the flows associated with a 5-year storm. My hunch is that I know for a fact it won't do the 100-year storm and it won't do the 50-year storm, but a bioretention cell can do the 5-year storm from the site that drains to it, okay? Because we can do a 5 to 10-year storm in North Carolina, and I don't think they're going to be that different because of our hurricane hits. All right, make sure everyone good about that. So the ability to mitigate a larger storm from a class 1 city in India is probably pretty good. Now, underdrains are required in bioretention cells as if the underlying soil has an infiltration rate that is relatively low. Now, 50 millimeters per hour is still, but do you remember what I talked about with permeable pavement? That you might start with a 50 millimeter per hour infiltration rate, and then you build it. And as you build it, you compact the underlying soils. Okay? Even though you don't want to, there's really no way you can't. The bucket comes in there, it scrapes along the bottom. You're just compacting it. All right. And so while 50 millimeters per hour is a pretty good infiltration rate, by the time you're done digging, that infiltration rate might be 10 millimeters per hour. Okay. Which is okay, but not as good. All right. And so basically, what the state of North Carolina says is if your starting infiltration rate is anything up to and less than 50 millimeters per hour, you need to put an under drain in. Okay. But the type of under drain you put in is going to help influence and improve your infiltration. And it's that upturned elbow you see diagrammed on this graph. Okay. You guys see that up there? About a half a meter. You want about a half a meter, a little bit less, 0.45 meters. You can submerge up to until the final 0.45 meters. All right. So this is what I'm talking about. You imagine the water comes in, and this device is called internal water storage. Basically, sets the water level inside at the nape or the bottom of this pipe. The ground if there's not a liner. That's what this is about. All right. And I can show you an example of one working. You can see the pipe coming out. There's a stand pipe coming up. The water flows up and out. And that's how this thing called internal water storage works. We require it to be used in North Carolina. And I'll explain why it's not just recommended, but required. First of all, we've studied it. Sandy clay loam underlying soil, different sizes of internal water storage volumes, monitored for a year and then another year. And you're looking at putting 70, 75, 80% of the water into the ground, okay? or into the atmosphere if you use internal water storage. That's really good. All right. We did a study where we had a relatively tight soil, hydrologic soil, group C, with 0.9 meters of media with an internal water storage. I want everyone to look right here at the amount of water that was pushed into the ground or exfiltrated, right? So we modeled, this is what we got. We then went ahead and modeled it again, this time without internal water storage, meaning that the pipe just came out. We weren't forcing any ponding, all right? Take a look at the purple lines. 20 to 25 is going into the ground. If you modeled it without internal water storage, it's 2.5 to 3 is going into the ground, all right? So in other words, you increase your infiltration by a factor of 10 by adding a T to your pipe, to your under drain. And it doesn't cost like $100. That's a really good return on your investment. All right. So that's why we require it. Another question that gets asked is, hey, how much media do I need to have here? Well, the answer is, what's the academic's favorite answer? If they say, how much blank do I need? What's their favorite answer? Remember this one, y'all. You can use it. It depends. It depends. Now, be prepared for the professor then ask you on what, all right? But it depends. Now, where are pollutants removed? Pollutants are removed from the surface, TSS. Phosphorus is removed at the surface and by the media. Temperature is reduced as the water hangs out in the media. Nitrogen is reduced as the water hangs out in the media. Pathogens are reduced at the surface because that's where they're captured and exposed to sunlight. Reduced in the media, generally towards the top. Oils and grease are reduced at the surface where the water is captured. We talked about some of these things on Tuesday, right? So how deep does your bioretention cell need to be? How much media do you need to have if you're trying to remove sediment? How much media do you need to have for sediment removal? Hardly any, right? Because as soon as the water filters through the media you have, the sediment that's been brought in is captured, right? But for temperature, if you're trying to cool the water down, then you actually need to have the media be deeper, all right? So how much media you need to have is wholly dependent on what pollutant you're trying to remove and what hydrologic goals you're trying to meet. The more media you have, the more water you can infiltrate into the ground. All right. So this is what we have provided, folks, because in North Carolina, most of the time we promote nitrogen removal, then the default depth that we have is close to a meter. Okay. Remember, we operate in feet rather than meters. So we would use 0.9 meters rather than 1 whole meter, but let's just go ahead and make it even. So 1 meter is what we use to remove nitrogen and phosphorus. That's the default. But if I'm just trying to get rid of pathogens, I don't even have pathogens up there. If I'm trying to get rid of pathogens, I could get away with 0.3, 0.4 meters of media, and I could kill the pathogens fine. Everyone on board with that? Think about what pollutant you care about, and you can adjust the bioretention design to meet that pollutant's removal. But I'm going to reiterate something I said just a bit ago. If you have hydrologic goals where you're trying to put more water into the ground, then you actually want more media. Does that make sense? The more opportunity you have for water to sit in an internal water storage zone, the more internal water storage you've got, ready for this? The more infiltration you're going to get. All right. Just remember that it's depending upon the goals you are trying to. By the way, if anyone ever cares, this is my all-time favorite journal article. I know it's been 13 years and I haven't had one that is better than this one. This is my favorite one. And it's I was the lead author with my buddy Dr. Davis and Dr. Traver. But you'll understand. And the thing is that that article, almost everything I share in it is still pretty good today. It's kind of cool. All right. So that's my favorite article. Maybe some of you someday will be able to say the same thing. If you get it, I hope you find it well written. I don't know. Okay. I'll skip. You can't, I will say you can have grassed covers. Okay. You can have turf grass associated with your bioretention. I'm not going to go into all the details, but you can have turf grass. It is particularly good for pathogen removal. More people will tend to use the tree shrub mulch design because it meets more landscape requirements. But a lot of people, if they can get away with turf grass, they will because it's cheaper. All right. Grass, you can have trees and shrubs, all that works. All right. Now, a little bit about the media. The media, you have to be very careful with its use. All right. For a long time, well, for like 5 years, 5 to 10 years, when media was going in, people were like, just make sure it is sufficiently coarse so that the water passes through. All right. And so people would actually choose media based on, like, the media composition would be based on what you wanted the infiltration rate to be. So if you were trying to have you bump up in your media, would you bump up, would you add more sand or add more silts and clays? You got it, silts and clays. Because if you add silts and clays, you're driving the infiltration rate of your media down. Why do you want a lower media infiltration rate? Because you need more contact time. So the longer the water's in there, the more nitrogen is going to get removed. All right. Do you see how it all kind of makes sense? What happens is it gets into some guidebook and people write down and they don't understand where these things come from. And then they feel like, oh, well, we got to make sure it's 25 to 50 millimeters. But if you're just trying to remove sediment, you can let it be 150 millimeters per hour. No big deal. You're just trying to get rid of sediment, trying to get pathogens, ditto. It's fine. Just you want your media not to be so high that grass can't grow or plants can't survive. I mean, there is an upper threshold for that, but anyway, that's where it comes from. So North Carolina, we worry about nitrogen, right? Because we worry about nitrogen, we tend to spec a lower media infiltration rate, or percolation rate, which means we have a little bit high, not a high amount, not a big amount, but a relatively higher amount of silts and clays in media mix. That's how all of that really, it's like Doctor Strange. By the way, any of y'all like Marvel comics? Do you think that I could have been Doctor Strange thank you for the media. I was going for that, but then my daughter's like, Daddy, I'm going to be a shark and I need you to be a fisherman. And you know what any good father would say to his daughter who asked him to be a fisherman? Yes, love. That's correct. If she wants me to, that means she needs me to hang around with her. I love it. Okay. Now, the next thing is a little bit tricky. If you're trying to remove phosphorus, you want to make sure that there's not a lot of phosphorus in your media already. You might think, well, okay. Where do you think most of the soil particles or the media, where do you think it's most typically harvested from? Where does it come from? Where do they? Another way to look at it, what type of land is most often converted to urban land? Is it forestry? You're right. What is it? Agricultural. And so people are like, well, I'll just take some of this nice agricultural soil that looks mostly sandy with a little bit of clay and silt, looks pretty good. I'll harvest that and I'll put it into my bioretention device. What has that farmer been doing on that soil for the last, I don't know, generation, 2 generations? Regent, whatever. What have they been doing to it? To get plants to grow, they've been fertilizing it. They may be using cow manure, all right? They may be, buying nitrogen and phosphorus off the shelf and putting it down there. The point is that there's a fair amount of phosphorus already word baked in, okay? A lot of phosphorus already present in agricultural soils. This phosphorus doesn't really go anywhere, all right? It just kind of builds up. So let me ask you this. Do you think it's a good idea to use soils that are high in phosphorus, stick them into a bioretention cell, and then flush a bunch of water through it, and then hope to remove more phosphorus? I'm like, that sounds like a great idea. You're right. No hands went up. You know why it's not you know why? Because it's not a great idea. All right, I'm going to show you a real quick example of that precisely happening. I studied this site in Greensboro. The soils in Greensboro came from a farm field. I studied a site in Chapel Hill. The media used in Chapel Hill came from a sand mine. The phosphorus removal from Greensboro was not that. We added 240% more phosphorus than came in. Now, the phosphorus can't magically appear. What we were doing is we were stripping phosphorus from the ag soil and discharging it. If this was the only site I studied, we would not have used bioretention in North Carolina. But I also happen to be studying the site in Chapel Hill because it was not farmer soil. We took out 65% of the phosphorus. And so when we saw those data, we're like, you know what? There's hope. So we tried it again. We tried again with a low phosphorus count, and we got about 50% phosphorus removal. So long story short, if you are trying to remove phosphorus with your bioretention cell, you are going to want to reduce or keep very low the embedded phosphorus in that media. Now, how do you know? What you want to do is you take soil samples from wherever you're getting your media, and you send it to a chemistry lab, a soils lab. I cannot imagine that there aren't any of those in India, because to optimize crop yield, you do have to know what your phosphorus soil content is, all right? So larger farmers are going to know, especially in this world of precision agriculture, they need to know how much phosphorus is already there. So there are labs that will study them for farmers. So your job as an engineer will be to figure out where those labs are, all right? And then align your media source with testing from that to make sure it's sufficiently low phosphorus before you put it in to the ground. Unless, of course, you just don't care about phosphorus. Can I tell you a place that does not care about phosphorus? They literally could care a rat's rear end about phosphorus. It's a place all of you have heard of, New York City. They don't care. You know why? Because when the water leaves New York City, do you know there's no barrier islands there? There's no estuary. That phosphorus literally gets diluted and nobody cares. Do you know what New York City cares about? New York City cares about one thing. From a stormwater perspective, they care that they don't have too high a flow rate so that their wastewater treatment plants I don't know. Did I just describe Bombay or Mumbai? I might have. Did I just describe Chennai? I might have. I tell you what I didn't describe. I did not just describe Bangalore. because you got lakes. You got lakes, you're going to care about phosphorus and nitrogen. But if you're discharging right into the Indian Ocean, yeah, you probably don't care. You don't have to care. You can care if you want to. Bangalore, you need to care. Delhi, you need to care if you worry about the quality of your receiving waters. But when I heard that Bangalore had all those lakes, I'm like, oh yeah, they need to care about nitrogen and phosphorus. Do you see why stormwater management needs to be, we'll call it regionally motivated, right? There's going to be things that you're going to care about here that you're going to not care, or they're going to be different from what you care about in Mumbai. There's going to be stuff in Delhi that's going to be different from Calcutta. There's going to be stuff different in Chennai as Bangalore. They're all going to be different. So hopefully all of you are taking good notes and you're like, okay, I'm working in Bangalore. I need to design my bioretention cell to get rid of nitrogen and phosphorus. And it's going to probably look different from one that maybe they're designing in Chennai where you not really care about nitrogen and phosphorus. Everyone on board with that? I know it's a little soapboxy, but I want you all to do the best jobs you can. You paid good money. You invested your time. Let's make it worth it, right? Mechanical compaction. You actually want to avoid it because what happens when you mechanically, let me see if I can get, yeah. I'm going to come back to a slide in a second. But what happens if you mechanically what's going to happen? What's going to happen to the infiltration and then percolation rate of that media? It's going to be damaged. It's going to get lower. All right. So you got to be careful about that. So one of the things that they actually recommend is that you, after you plant media, is that you water it in or you even walk it in. Look, I've seen a lot of maintenance staff on campus here. It's not that much. I could see that happening where you put a bioretention cell in, and you get a group of dudes, and they go in and they do like this. All right. It might even be fun. You might even be able to moonwalk. I don't think it'd be easy to moonwalk, to be honest. Do you guys even know what the moonwalk is? Michael Jackson. Look that up. Look, you didn't just come for engineering, right? We came for a little pop culture. We'll call it historical pop culture, 1980s style. All right. I won't get into that. Okay. Last thing I want to talk about. We're doing great. Last thing I want to talk about, or second, there's two more things, is the planting plan associated with bioretention. Please hire someone that knows what they're doing. Now, the good news about bioretention plants as compared to green roofs, like green roofs, it's a desert, right? A green roof media, 10 to 15 centimeters of dry media, rock-based media, that's a desert. That's hard to get most plants to grow. Bioretention, different story. You're going to have a meter, or at least 2/3 of a meter of media, right? It's going to have silts, clays, mostly sands, but some silts and clays in there. There is water capacity in there for many, many plants, all right? The bigger issue is just trying to make the plants look good, all right? And also understand that they will evolve with time. So you get an aesthetic that fits in with the building or the landscape that you're trying to treat. We actually promote a relatively open design with low shrubs and a few trees because we believe that that particular system is better for pathogen die-off. All right, if your system is like this, it's better for pathogen die-off. All right, but again, the most important thing I can share with you is go to somebody who understands to help you select what goes in. All right, mulch. Bioretention cells, by and large, should have mulch. It doesn't have to be wood mulch. It can be, but wood mulch floats. We actually have begun to recommend a gravel mulch, a light colored gravel mulch, all right, that the plants can survive in. Again, there is a bit of an albedo consideration, all right? And again, you can also go to plant experts and say, look, I'm going to have a white gravel mulch. Here's one that we have at my university close to my office, a white gravel mulch. What can grow in it? All right. And they can tell you. You tell them that, and you tell them how moist it is, they can give you the answer. There should be clean out pipes. The advantage of clean out pipes, of course, is that you can check on how well they're working. It's a way to do maintenance if you need to. All right. We use concrete donuts to try to protect the green to protect them so they don't get broken. All right. So that's bad. That's fine. And then the last but not least, with bioretention, we are strong proponents of using forebays to clean the water before it goes to the bioretention cell. You can't always do that because sometimes the water enters bioretention via sheet flow, and that's okay. But if you have a defined entry point, having a little forebay in there where the sediment can accumulate so that it doesn't spill over into the rest of your media can facilitate, and that little forebay helps facilitate maintenance, man, that's a good idea. Okay. Not that dissimilar from what I talked about yesterday with ponds. Now, the last thing I want to share with you are you can learn a lot from mistakes. So I'm going to share with you a few mistakes. We were really excited about this set of bioretention cells. You see them going in here, they're pretty cool. You might also observe that this one here has got water in it. Well, the reason is we did not soil sample all of the places where we wanted to put bioretention. We took 4 soil samples to make sure that the underlying soil was sufficiently permeable. All right. So this one site, the answer was no, it wasn't permeable, but we didn't pick up on that. Okay. So, and we didn't design for that. So in the end, this was actually a very costly fix because we had to retrofit a pipe in here and tie it into a concrete structure right over here. All right. So just be mindful that before you decide how to design your bioretention cell, understand underlying soils are at the location of the cell. All right. Here's one where the bioretention cell died. It was DOA, dead on arrival. Why? Because the media was bad. It had too many fines, too many clays in it specifically, and it was too expensive to fix. So instead, what they did is they knocked a hole in the bottom of the outlet structure. So what did they turn this bioretention cell into? It was something we talked about yesterday. I don't see ponded water, so what does it not necessarily have to be? I think I hear a dry pond. That's right. They turned the bioretention cell into a dry pond. It was a very expensive dry pond. This is a killer for bioretention. What's the color of that water suggest to you? That there's sediment in the runoff. Bioretention cells can handle a little bit of sediment, but if you expose a lot of sediment, I don't have a great picture of it. I'm sorry. But if you expose a lot of sediment, then it will crust over on top of the bioretention cell and the water will not drain through it. This might be the most important thing I'm going to share with you today about bioretention and the Rookery campus. There are some spots that are going to be free and clear of sediment, but not every spot is. Okay? And so maybe to your bioretention cell does not have if you have a little bit, it's okay, but you don't want a lot of exposed sediment because it will clog the process. If you let it go crazy, all right, this is what happens in Singapore where they have a lot of water, okay? You can start having issues with animals living in there. You guys don't really have any things that can kill you here, do you? Do you? What's your snake situation? You have a lot of venomous snakes here? Where do the cobras live? You have any cobras? No? Yeah. Cobras lived in this one. All right. And the maintenance staff was like, I said, what do you do when you see a cobra? We're like, we turn around and walk out. I guess that makes sense. All right. But critters do get in there. We even have it, and maybe it's a big issue, maybe not, but we have people that will live in bioretention cells, right? Because they're dry unless it's raining. So during the dry periods, you kind of tuck in behind the vegetation. We found mattresses. People. Okay. And maybe some people are fine with this look, all right? But not everywhere is. And certainly in ultra urban areas, this may not fly. This might be a little bit of a problem. All right. I think bioretention, as much as we do, is the practice works very, very well at removing pollutants. And remember, doing this and doing this. Do we have any questions? I promise to get you guys out of here by 1 o'clock. That's about as close as it gets. Yes. Hold on, I'm coming to you. The under drain? Okay. Say it one more time.
The drain increases the infiltrate.
Okay, yeah. So when you have an upturn in the under drain, you basically force water to pond over the bottom of the bioretention cell. The bottom of the bioretention cell, we do not line, okay? Unless we have an issue with really polluted soils underlying it, but we don't line it. And so what ends up happening is if you force this much water, or in some cases like this much water on top of an infiltrating plane, you have a higher head, you just have more infiltration. And that's how it helps. Any other questions? I'm glad you asked, because I want to make sure everyone understands that. The most popular urban practice in North Carolina, not suburban, not peri-urban, the most popular peri-urban is the pond. The most popular urban practice in North Carolina is bioretention, okay? Because it does improve the hydrology and it's really good at removing pollutants. It's better than any practice I shared with you. The best practice, even wetlands, the best practice at removing pollutants, assuming it's working right, is bioretention. Because it is a vegetated area, so you get filtration, but because it's vegetated, you also get a lot of biological activity, which helps kill pollutants. So that's why bioretention is so good, and that's why we use it as much as we do, all right? Any other questions? All right. Dr. Venarasi, Professor Venarasi, Professor VR. Like, don't call me that. What would you like all of our scholars to know between now and test time? I told you guys we get at least an hour. I want you guys to have at least an hour for lunch.